Continuation with the previous class where we have discussed about the glycosaminoglycans and also the glycoconjugates, mainly proteoglycans. So in glycosaminoglycans, we have discussed about the structure and properties of hyaluronic acid, chondroitin sulfate, keratan sulfate, dermatan sulfate, and heparan sulfate. Then in glycoconjugates, we have discussed about the proteoglycans, where the glycosaminoglycans are covalently linked to the proteins, and these proteoglycans have various functions. I think I will be recording a separate lecture on different types of proteoglycans so that you can see later. So in today's class, we discuss about the classification of amino acids and properties and amino acid titration curve. And you can see here in this module, we have classification of amino acids and structure and biological importance of amino acids, acid-base chemistry and stereochemistry of amino acids, and different structure and function of different proteins like keratin, collagen, silk fibrin, and globular proteins such as hemoglobin and myoglobin. So in order to understand the structure and function of hemoglobin and myoglobin and other proteins, first we need to understand the properties of amino acids, because proteins are built with the amino acids. So we know that amino acids are the carboxylic acids containing amino group. You can see here they are the carboxylic acids containing the amino group, and in most of the amino acids, the amino group, this is attached to the alpha carbon. This is the carboxyl group and this is the alpha carbon. So you can also see here alpha carbon atom, that is the next to the carboxyl group. Hence they are called as the alpha amino acids. And coming to the medical and biological importance, and these amino acids are the building blocks of proteins first. And in addition to this, when they are also the precursor, the amino acids are also precursor for the hormones, purines, pyrimidines, porphyrins, vitamins, and biologically important amines like histamine. And some of the proteins are also the hormones. This we will see later, and some peptides are required for the detoxification reaction, and some peptides are the neurotransmitters, and the amino acid proline protects the cell from the free radical induced damage, and some peptides are also involved in the regulation of the cell cycle and apoptosis. And some peptides of vertebrates and invertebrates acts as antimicrobial agents, and some peptides are enzyme inhibitors. And this you can see here that the tyrosine, the amino acid tyrosine, is the precursor for the hormone epinephrine. The tryptophan is precursor for the melatonin. In the same way, you can see here the tyrosine is the precursor not only for the epinephrine, and it is also precursor for the dopamine and norepinephrine and so on. And the histidine is the precursor for the histamine. It is a vasoactive amine, and that means these amino acids are not only the part of the proteins, they are the source for the formation of many important molecules. And you can also see here the dopamine and the norepinephrine, serotonin. All these are the biogenic amines of all these are synthesized from the amino acids. And gamma aminobutyric acid is again a neurotransmitter, and the glycine and gamma aminobutyric, they are the inhibitory neurotransmitters, and glutamate and aspartate also the excitatory neurotransmitters. And as I already said that the proline is involved in the protecting the cell from the oxidative mediated damage, and some of the peptides are the enzyme inhibitors. For example, HA protease inhibitors. You can see here all these are peptides in nature. And as I said that some Peptides are also the neurotransmitters. For example, enkephalin is a neurotransmitter. And coming to the classification, all these amino acids are classified in various ways, but more than 200 amino acids have been identified in nature, but only 20 amino acids are only present in the proteins. They are also known as the common amino acids or the standard amino acids. And these 20 amino acids that are present in the proteins are classified into different categories, and mainly they are classified into three groups based on the reaction and solution. They are the neutral amino acid, acidic amino acids, and basic amino acids. And in addition to this, the amino acids are also classified in various ways based on the side chain and the ring structure present. They are classified into 7 major classes. And the first one is the amino acids having aliphatic side chain. And here the glycine, alanine, valine, leucine, and isoleucine. All these have you can see here, they have the aliphatic side chain. So this is the side chain. This is the basic structure of the amino acid. It has a carboxyl group, one side amino group, and this is the R group. This is the side chain. So they differ from one another only at the R group. And so all these are the amino acids having the aliphatic side chains. And then second one is the amino acids having the hydroxyl group containing side chain. For example, here serine, threonine, and tyrosine. You can see here all these three amino acids have the hydroxyl group. Of course, tyrosine is the aromatic amino acid, but it has the hydroxyl group. That's why it is also brought under this category. So the serine, threonine, tyrosine, all these amino acids have the hydroxyl group in their side chain. And third class is the amino acids having the sulfur atoms in the side chain. For example, methionine, it has the sulfur and the cysteine, and these two amino acids have the sulfur in the side chain. Of course, the cysteine, it is the dimeric form of the cysteine, and it is not listed in the among the 20 amino acids. It is the dimeric form of the cysteine, is known as the cysteine. And then fourth group is the amino acids containing the acidic groups or their amides. So two amino acids have acidic groups. One is aspartic acid. It has one extra carboxyl group, and second one is the glutamic acid. In addition to this, the amide form of this acidic group is asparagine. So in the sense here, the amino group is substituted, and the glutamine, these are the amide form of the acidic amino acids. Then amino acids having the basic groups. So we know we have only three basic amino acids. One is arginine, lysine, arginine, and histidine. These are the three the basic amino acids. And you can see here in acidic amino acids, we have one extra carboxyl group in addition to the alpha carboxyl group and the amino group. Whereas in basic amino acids, we have one extra amino group in addition to the alpha carboxyl group and the amino group. So these are the basic amino acids. Then coming to the aromatic amino acids having the aromatic side chain. So we have phenylalanine, tyrosine, and tryptophan. These three are aromatic amino acids. They have the aromatic side chain. Then the amino acid. So we have total 20 standard amino acids. As I already said that we have 20 standard amino acids that are present in proteins. Among these 20 standard amino acids, 19 are amino acids, one is amino acid. In the sense, it has all 19 amino acids have the primary amino group, and whereas here this has the secondary amino group. In the sense, this nitrogen is bonded to 2 carbons. So this is here the amino acid is the proline, and the proline plays an important role in the collagen structure that is in the form of hydroxyproline. This we will see later. So the proline is the amino acid which has the secondary amino group instead of all other amino acids have the primary amino group, whereas here the proline has the secondary amino group. That's why it is called as the amino acid. So these are the ways the amino acids are classified based on the side chain and the ring structure into 7 classes: amino acids with aliphatic side chains, the hydroxyl group containing side chains, and the sulfur group containing side chains, and the acidic groups are their amide containing side chains, and the basic group containing side chains, and aromatic ring containing side chains, and as well as the amino acid. And as I said that based on the reaction in the solution, these amino acids are also classified into 3 classes, as I already said, that acidic, basic, and neutral. So we have only 2 acidic amino acids. One is aspartic acid, second one is the glutamic acid. They have one extra carboxyl group on the side chain. And in the same way, we have 3 basic amino acids. One is arginine, lysine, and the histidine. And the remaining amino acids are the neutral amino acids. So this is the these are all the 20 standard amino acids you can also see here, and they differ from one another only the side chain. You can see here the side chain has been shaded with a different color. You can notice that they differ from one another only at the side chain. And so this is about the classification of based on the reaction. Then all these amino acids have the 3-letter code as well as a single letter code, and you should understand the 3-letter code and the single letter code for all these amino acids. For most of the amino acids, the 3-letter code is the first 3 letters of the name of the amino acid, but the single letter code is also for most of the amino acids starts with the first letter of the amino acid, but for some amino acids it is different. For example, arginine, the single letter code is R, asparagine N, aspartic acid D, and the glutamin Q, and glutamic acid E, and lysine K, and the phenylalanine F, and the tryptophan W, and so on. For other amino acids, the single letter code is nothing but the first letter of the amino acid. And then coming to these amino acids are also classified based on their the number of carboxyl group and the amino group present. One is monoamino monocarboxylic acid. For example, all neutral amino acids have only one carboxyl group on amino group, and monoamino dicarboxylic group, in the sense they have one amino group and the 2 carboxyl group like glutamic acid, aspartic acid, and so on. And third classification is based on their nutritional importance. So based on their nutritional importance, these amino acids are classified into essential and non-essential amino acids. And these essential amino acids these essential amino acids are not synthesized in the body, hence they need to be supplemented in the diet. And these essential amino acids you can see here. I have given a short cut to remember these essential amino acids. Mat will fly. So M stands for methionine, A stands for arginine, T stands for tryptophan, next T stands for threonine, V stands for valine, I stands for isoleucine, and another L stands for leucine, and P stands for phenylalanine, H stands for histidine and lysine. So methionine, arginine, tryptophan, threonine, valine, isoleucine, leucine, phenylalanine, histidine, and lysine, all these are essential amino acids. And histidine and arginine considered as a semi-essential because the human body can synthesize these amino acids to some extent. And lack of these amino acids in the diet give rise to the growth failure. And coming to the non-essential amino acids, we have the alanine, glycine, serine, tyrosine, glutamate, glutamin, aspartate, asparagine, cysteine, and proline. These are non-essential amino acids because they need not be supplemented in the diet. They are synthesized in the body, and you can also see this the way to remember AGST, GG, AA, CP. So alanine, glycine, serine, tyrosine, glutamate, and glutamic acid, and glutamin, aspartic acid, and asparagine, and cysteine, and proline. These are non-essential amino acids. And then coming to the as I already said that more than 200 amino acids have been identified, but only 20 are present in the proteins. They are known as the standard amino acids. But in addition to the standard amino acids, many amino acids that are not present in the proteins, but they have the various roles in the cell. For example, ornithine and citrulline. You can see here this ornithine and citrulline, but these are unusual amino acids. Unusual amino acids. They are not present in the protein, but they have various functions in the cell. Ornithine and citrulline and arginosuccinic acid, these are the intermediates in the urea cycle. I think we have already know this one. And beta alanine is the part of the coenzyme A. This is the beta alanine, and this is the part of the coenzyme A. Taurine is a part of the bile acids, and taurine is the part of the bile acids, and you can see here. Then the gamma aminobutyric acid is a neurotransmitter. Mono and diiodotyrosine are the precursor for the thyroxine. You can see here, this is the thyroxine, and thyroxine you can see here. This is the all these MIT, DIT, monoiodotyrosine, diiodotyrosine, all these are the precursor for the formation of the thyroxine and reverse T3, and also triiodotyrone. All these are the thyroid hormones, so they are derived from the MIT, DIT, diiodotyrosine, monoiodotyrosine, and so on. So that means the diiodotyrosine and monoiodotyrosine, all these are the precursor for the thyroxine. And of course, pantothenic acid is a water-soluble vitamin. Homoserine is intermediate in the methionine metabolism, and homocysteine is also intermediate in the methionine catabolism. And so coming to so in addition to this, some non-standard amino acids, mainly hydroxyproline and hydroxylysine. These two are very, very important for the structural function of the collagen. If you see the collagen structure, you will understand that the hydroxylysine and the hydroxyproline is present in the collagen. And you can also see here, this is the lysine. Lysine is converted into the hydroxylysine, and the proline would be converted to hydroxyproline during post-translational modification. And this conversion of proline to hydroxyproline and lysine to hydroxylysine is catalyzed by the hydroxylase enzymes. These hydroxylase enzymes, prolyl hydroxylase, lysyl hydroxylase, they use vitamin C as a cofactor for this reaction. And this hydroxyproline and the hydroxylysine is important for the collagen structure. And deficiency of the vitamin C leads to the insufficient formation of the hydroxyproline as well as hydroxylysine. As a result, the collagen that affects the collagen synthesis. That's why deficiency of the vitamin C leads to the scurvy or the and so on. So then coming to the properties of amino acids, and we know that all amino acids except glycine, we have already seen that except glycine, all other amino acids have one or more chiral centers, and so that's why they exhibit the optical isomerism. And so in all these amino acids, you can see here, if the amino group is present on the left side, then it is the L amino acid. If it is present on the right side, then it is the D amino acid. You can also see here, and only L amino acids are naturally present in the proteins, but not to the D amino acid. And of course, D amino acids are also present very rarely, but naturally only L amino acids are present in the proteins, but not the D isomers. But the D isomers also present in some cases, like in some antibiotics, like the valinomycin contains the D-valine, and also the peptidoglycan, as we have already said in the previous class. It is a NAG&NAM polymer, and this one NAG&NAM polymer is connected to another NAG&NAM polymer through a short peptide, and this short peptide also has the D-alanine. That means the proteins, only L isomers are present in the proteins, but the D amino acids are also present in special structures. and but they are not naturally present in the proteins. And coming to the acid base or the charge properties of amino acid, since the amino acid has both amino group and the carboxyl group, so they are called as ampholytes or the amphoteric substances. In the sense, in the presence of alkali, the amino acid acts as an acid, whereas in the presence of acid, the amino acid acts as a base. In the sense, according to Bronsted-Lowry theory, proton donor is acid and the proton acceptor is base. So in the presence of alkali, the amino group loses the amino group of the amino acid loses the proton, whereas in the presence of acid, the carboxyl group against the proton. So that's why the amino acids can release the proton, can also accept the proton. So that's why they act as an acid and bases. And if you see the charge properties, the amino acids have the zero charge at the isoelectric point, and below the p isoelectric point they are positively charged, whereas the above the isoelectric point they are negatively charged. For example, this is the isoelectric pH when the below the isoelectric pH, in the sense where the H plus ion concentration is more, this H plus ion combines with the COO minus and it becomes COOH. Therefore, the net charge is then the amino acid becomes a cation, and so above the isoelectric point where the OH minus again pulls one more proton from the NH3 plus. As a result, it becomes NH2. Therefore, the amino acid becomes anion. So that means at neutral pH, in the sense that isoelectric point, the amino acid exists as a cation. Below the isoelectric point, it exists as a cation. Above the isoelectric point, it exists as an anion. And as I already said, this amino acid can release the proton, can also accept the proton. That's why they act as an acids and bases. And the isoelectric point is the pH at which the amino acid has a net zero charge. That is the isoelectric point. Different amino acids have different pH values. You can see here. But the point you need to remember here is that for basic amino acid, the pH value lies on the basic side, whereas for acidic amino acids, you can see aspartic acid, the pH value lies on the acidic side. And for the neutral amino acid, the pH value is almost close to the from 6 to 7 in between, 5 to 5.5 to 7 in between. And then coming to the buffering action of amino acids. Since the amino acids are ampholytes, as I said, that they can release the proton, they can also abstract the proton. So depending upon the condition, so they act as a buffers in the buffers. And another important property of amino acids is the absorption of UV light. So amino acids do not absorb light in the visible region, but the aromatic amino acids, mainly tryptophan, tyrosine, phenylalanine, they have the aromatic rings and with many double bonds, and these double bonds are responsible for absorbing light in the UV region, that is at the 280 nanometers. So that's why, so when we can also measure the protein concentration at 280 nanometers, because the proteins also contain the many aromatic amino acids. These aromatic amino acids present in the proteins can absorb light at 280 nanometers. And you can also see here all these amino acids, phenylalanine, tyrosine, tryptophan, these three only absorb light in the UV region, that is around the 280 nanometers. None of the remaining amino acids out of 20 standard amino acids can show the absorbance at 280 nanometers because they don't have the aromatic ring. And then coming to the titration curves of amino acid, we know that as we said that amino acid can act as an acid as well as the base, in the sense they can release the proton, they can also abstract the proton. So that's why they have the both acidic and basic properties. And so all amino acids have ionizable groups. One is a alpha amino group, another one is a carboxyl group. And when we titrate the amino acid with either acid or the base, we can find out the pKa value and the Pa value and also the buffering region. So we know that in any titration curve, generally we determine the strength of unknown using the strength of known. But in amino acid titration curve, we titrate the amino acid with either acid or base, and we measure the pH continuously. And when we construct a plot that is versus the alkali added versus the pH, we get a different shape of the curve. This is the amino acid titration curve, and this amino acid titration curve is useful to determine the PA value of the amino acid and also the PK value of the amino group as well as the carboxyl group. And also we can also determine the what is the buffering region where the pH is stable. And so that means the amino acid titration curve is used to determine the PA value and the PK value, and also to determine the buffering region of the buffering region. And so when we titrate, I will just briefly explain this one. So when you, for example, when you take the glycine, when we take a glycine, and when the alkali sodium hydroxide is added, and the pH is measured continuously using the pH meter. So at extreme lower pH, for example, for the sake of understanding, assume that at extreme lower pH, the glycine exists as I already said, that glycinate cation, in the sense the carboxyl group is protonated, amino group is also protonated, therefore this exists as a cation. So when we slowly add alkali NaOH, NaOH, and initially when we add alkali OH, this OH minus pulls the proton from the alpha carboxyl group, and as a result the water would be removed, and the CO becomes CO minus. That means glycine becomes glycine deuterion, or it is also glycine deuterion, because here it has only +1 charge and here it has only 0 charge, and further addition of alkali, the OH minus also pulls the proton from the amino group. As a result, it becomes NH2 again. So therefore, the net charge is -1. So that means at the extreme lower pH, the glycine exists as a glycinate cation, and addition of alkali, as the alkali is added, initially the alpha carboxyl group loses proton and it becomes the glycinate glycine deuterion, and further addition of alkali, the amino group also loses proton and it becomes a glycinate anion. So that means, and as we during the process of this titration, when we measure the pH and when we construct a graph between the moles of OH minus added versus the pH, we get this type of titration curve. And in this titration curve, and we can see that there are 3 flat zones here. This is the one flat zone, and this is the another flat zone, and the midpoint of the flat zone is nothing but the pK, and the midpoint of the first flat zone is the pK1, that is due to the alpha carboxyl group, and the midpoint of the second flat zone is the pK2, and this is because of the alpha amino group. And you can also see here, and this is the PA, in the sense where the net charge of the glycine is 0. So that means we can understand that by using this the titration curve, we can determine the PK value. We can also determine the PK1 and PK2, and for the, since the glycine is a neutral amino acid, the PA is equal to PK1 plus PK2 by 2. So that will give you, this is the, so this is, so 2.34 plus 9.60 divided by 2, that is equal to 5.99. So that is the PA value. And at the PA, the net charge on the glycine is 0, and the glycine exists as a deuterion. So this is the way we need to understand the glycine. And of course, it is the same thing for the alanine, and this we no need to say explain. Then coming to the, and I just wanted to explain one thing here, that so in order to understand the the behavior of the amino acid in the titration curve, we use a Henderson-Hasselbalch equation. So the Henderson-Hasselbalch equation, we know that is the pH is equal to pKa plus log the unprotonated form, that is based by protonated form acid, based by acid. So you can in the titration curve, so you can see in this titration curve, and the pK1 and the midpoint of the first flat region is pK1 and the midpoint of the second flat region is the pK2. So as I already said that at extreme lower pH, the glycine exists as a glycine in the cationic form, and so in the midpoint of the flat region, so the cationic form, and the deuterionic form both are present in equimolar amounts. In the sense, 50% of the cationic form and the 50% of the deuterionic form is present here. So when we apply the Henderson-Hasselbalch equation here, the pH is equal to pKa plus log base by acid. So here, so this is both, that is the protonated form and the unprotonated form both are present in the equimolar amounts. Therefore, so this component becomes 0. So therefore, the pH is equal to pK. So the pH at this point, is nothing but the pKa. So in the same way here. So that means the Henderson-Hasselbalch equation is used to understand the behavior of the amino acid and its net charge during the titration. So this is about in case of neutral amino acid, because neutral amino acids have only one amino group, one carboxyl group. Coming to the acidic amino acids, and we have here the glutamic acid, and you can see here. So when the glutamic acid is titrated with the alkali, and you see that how the ionization state of the glutamic acid changes. So as I already said that glutamic acid at extreme lower pH, the glutamic acid also exists as a protonated form, in the sense the alpha carboxyl group is protonated, alpha amino group is protonated, and the side chain carboxyl group is also protonated. So that means extreme lower pH, all the three ionizable groups are protonated. Therefore, the net charge is +1. You can also see here +1. And when the alkali is added, and initially when we titrate with the glutamic acid with the sodium hydroxide, and this alkali pulls the first proton from the carboxylic group, and as a result it becomes CO minus. Further addition of alkali, the side chain carboxylic group also loses a proton, and as a result again it becomes a CO minus. And further addition of alkali, the alpha amino group also loses a proton, as a result it becomes an H2. So if you carefully see here that at the extreme lower pH, the net charge on the glycine is +1, and when it is titrated with the alkali, initially the alpha carboxylic group loses proton, as a result it becomes CO minus. Therefore the net charge becomes here NH3 plus, here CO minus, so the net charge is 0, and further addition of alkali pulls the proton from the side chain carboxyl group. Therefore, the net charge is -1, here 0, here +1, and further addition of alkali, the amino group also loses proton. So therefore, the net charge becomes -2. So that means during titration, as the alkali is being added, and when we measure the pH also changes, and for glutamic acid we get this type of titration curve, and this you can clearly see here. So it has the 3 flat regions in this graph. You can clearly see here it has the 3 flat zones. This flat zone 1 is because of the carboxyl group, and flat region 2 is because of the side chain carboxyl group, and flat region 3 is because of the alpha amino group. In the sense, as I already said that here in the midpoint of the flat region 1, that is a pK1, and so at the pH at this point is pK1, and here the glutamic acid, the protonated form, and the these two forms are present in equal amounts, whereas here these two forms are present in that is deuterionic form as well as the glutamic acid with a -1 charge. These two forms are present in equal amount, and here these two forms are present 50-50 ratio. And when we apply the Henderson-Hasselbalch equation, we will understand the pH at that point is nothing but the respective pKs. And here pK1 is because of the ionizable group alpha, the carboxyl group. pK2 is because of the side chain carboxyl group. pK3 is amino group. In the sense, during titration, the more acidic protons would be lost first. So here, when we have three ionizable groups, so the alpha carboxyl group only loses proton first, then the side chain carboxyl group lost the alpha amino group. So in this case, in case of acidic amino acids, the pi is equal to the pKa values of two similar functional groups. Here, two similar functional groups are alpha, the side chain carboxyl group, and as well as the alpha carboxyl group. Therefore, the alpha carboxyl group and the side chain carboxyl group, so therefore the Pa is equal to pK1 plus pK2 by 2. So since the glutamic acid is a acidic amino acid, you can notice here that the Pa value lies on acidic side, that is around 3.25. So this is about the acidic the titration curve of acidic amino acid. Coming to the titration curve of basic amino acid, we have the lysine, that is a basic amino acid. So as usual, at extreme lower pH, all the three functional groups, carboxylic, alpha carboxyl group, alpha amino group, side chain amino group, all these are protonated. When the alkali is when it is glutamic acid is titrated with the alkali, when the alkali is added, first the alpha carboxyl group loses proton and it becomes CO minus. Therefore, the net charge becomes +1 from +2. Initially it is +2, it becomes +1. Further addition of alkali, the alpha amino group also loses a proton and it becomes NH2. So the net charge is 0. And further addition of alkali, the side chain carboxyl group also loses proton and it becomes a -1. And again here, the pK1 is because of the alpha carboxyl group, pK2 is because of the alpha amino group, pK3 is because of the side chain amino group. And in case of basic amino acids, the Pa value is equal to here the pK2 plus pK3 by 2, because as I already said that for basic amino acids, the Pa value is equal to the average pK of two similar functional groups. The two similar functional groups are alpha amino group as well as the side chain amino group. Therefore, the Pa is equal to pK2 plus pK3 by 2. So it has, you can see here, it is almost 10, and that means since it is a basic amino acid, its Pa value lies on the alkaline side. And so this is about this one. In addition to this, we have the another amino acid that is histidine. Histidine is also the basic amino acid, but it has you can see here that at extreme lower pH, all the carboxyl group and the alpha amino group and also the ring amino group, all these are protonated, and is added. First alpha carboxyl group loses proton. Further addition of alkali, the imidazole nitrogen loses proton, because the ring protons are more acidic compared to the aliphatic protons. That's why. So this loses proton, and you can see here. And further addition of alkali, the alpha amino group also loses a proton. So that means in this case, pK1 is because of the alpha carboxyl group, and pK2 is because of the side chain, the imidazole group, and the pK3 is because of the side chain amino group. And of course here it is written as pKr, and pK2 it is okay. In some textbooks you will see pK1, pK2, pK3, or pK1, pKr, and pK2. pKr means it is the pK value due to the side chain. And in this case, again, the PA value is equal to PKR plus PK2 by 2. So in the sense, this is the amino group, and this is also amino group. So the pK2, pKr plus pK2 by 2 by 2. So this is the way you need to understand the titration curves. As I already said, that titration curves in amino acid titration curves are useful to understand how the amino acid behaves, whereas the when we add alkali or when we titrate with alkali or acid, and in this we titrate either we titrate with acid or alkali, and we measure the pH. When we construct a plot between the volume of the moles of alkali added versus pH, we get a different titration, different shape of the titration curves. And another important point here, you need to understand that the shape of the titration curve, the shape of the titration curve will tell you that how many ionizable groups are present in an amino acid. For example, you can see here that if the amino acid has only two ionizable groups, in the sense one carboxyl group, one amino group, you would see only the two flat regions. And if the amino acid has three ionizable groups, you will see one flat region and another flat region and another flat region. In the sense, the number of flat region is nothing but the buffering zone, in the sense where addition of alkali has not much dramatic change in the pH. You can see here. So this region has a dramatic change in pH when you add alkali. When the alkali is added, there is a drastic change in the pH, whereas this region doesn't have much change in the pH. So these are the buffering regions. And so that means if the amino acid has three ionizable groups, you would see the three flat regions in the titration curve. If the amino acid has only two ionizable groups, you would see only the two ionizable groups in the titration curve. That means by looking at the shape of the titration curve, we can understand that how many ionizable groups are present in a given molecule. So, and as I already said, that this titration curve is useful to determine the pK values, to determine the PA values, to know the buffering region, and also to know the behavior of amino acid. So this is the importance of the amino acid titration curves. So this is the first part of the this topic. I think I will take one more. I will record one more lecture that is on the second part of this one, peptides and peptide bond as well as the protein structure. So this I will record the lecture. You can go through this lecture after some time.